So namaste. So today we are going to discuss one of the topic in the plant breeding that we call as somatic hybridization. So here in the plant breeding there are the various techniques. There are the various techniques are available to bring the variations in the biological population. Some are the conventional breeding techniques, some are the modern breeding techniques, some are the molecular breeding technique. So with the help of this breeding technique, there is a developed variations in the biological population can be possible. According to the variation, there is a development of the new character and allele can be possible. Based on the new allele, there is a development of the new variety in the agriculture crops can be possible. That's mean, so because in the basic objective in the plant breeding is to produce the better genotype, the better genotype than the existing one. So in order to make the better genotype, there are the various breeding techniques we have in the agriculture, especially we are having the some are the mass selection, some are the purine selection, some are the pedigree methods, some are the single seed descent methods, some are the backcross methods, some are the synthetic, some are the composite method, and some are the somatic hybridization. So based on this approach, there is a improved variation in the biological population can be possible, right? So here today basically we're discussing the synopsis of the somatic hybridization. In the somatic hybridization, sometimes it is also called as the wide hybridization. Is also called as the wide hybridization. The wide hybridization, or sometimes it is also called as the distant hybridization. It is also called as the distant hybridization. The distant hybridization. See, look at the board here. So every year we can expect very good number of the question in the case of the somatic hybridization. Either can be 15 or 20 marks question. Always can be possible in the case of the somatic hybridization. Somatic hybridization, sometimes it is also called as the wide hybridization or the distant hybridization. This means irrespective of the any plant species, hybridization can be possible in the case of the somatic. So in this cases we are taking the somatic cells. Somatic cells matlab we are having the protoplast culture. So protoplast culture will be isolated from the different plant species. After that there is a fusion of the protoplast is going to take place in the case of the somatic hybridization. So here in the case of the somatic hybridization, right, we're having these are the few examples we have. That's mean irrespective of the plant species, the hybridization can be possible in the case of the somatic hybridization. That is the one thing we say. So in this cases we are taking the protoplast culture one from the potato plant and protoplast culture from the tomato. So once after taking the both the culture, then fusion of the both the culture is going to take place for the development of the F1 progeny we called as tomato plant. So tomato is the one of the advancement, one of the achievement we made based on the somatic hybridization. Like that, so we have taken the protoplast culture from the brinjal and culture from the another culture from the tomato. Then according to that, in the F1 progeny we are having the bremato. Like that, even in the animal husbandry we are having some example we called as lion and donkey. Among the two species, then there is a hybridization compatibility can be possible for the development of the lion. Then we are having the horse and donkey for the development of the mule. So in these cases, right, so from both the culture, right, we are having the chromosome composition we called as we are having the nucleus plus cytoplasma. In both the parents we are having the nucleus plus cytoplasma, both will be inherited into the progeny we called as symmetric hybrid combination we say in general. Like that, they are the different culture. So this means irrespective of the any plant species, the hybridization can be possible in the case of the somatic cell, right? But in the sexual hybridization, right, cytoplasma is strictly contributed by the female parent. You have to remember, in the sexual hybridization, cytoplasma is strictly contributed by the female parent, but in the somatic hybridization, cytoplasma is contributed by the both the parents, parent 1 and parent 2. According to that, we're developing the improved varieties in the agriculture can be possible. So as of now, in the based on the somatic hybridization, presently at the experimental level, we're having the tomato experiment, another is the bremato. So this means irrespective of the any species, the hybridization can be possible based on the technique of the somatic hybridization. Is one of the technique in the molecular breeding. According to that, there is a variation in the biological population can be possibilities, right? So in this cases, right, see the somatic hybridization, we're having the different type of the somatic hybrid can be possible based on the taxonomic relationship. Sometimes it can be called as interspecific hybrid, right? At the species level, there is a protoplast fusion is going to take place for the production of the interspecific hybrid, and we are having that the genus level. At the genus level, we are having the interspecific hybrid. Then at the family level, you are having the inter-family or tribe level, we say. So based on the taxonomic relationship, now we are having the three type of the somatic hybrid can be possible. One is the interspecific, another is the intergeneric, another is the interfamily hybrid can be possible. That is the one classifications we have in the case of the somatic hybrid. Another one based on the chromosome composition. Based on the chromosome composition, right? So we said that from both the parents, there is a equal contribution of the nucleus plus cytoplasma is going to be inherited from the parent to the progeny in the case of the symmetric hybrid. In the case of the symmetric hybrid, there is a equal chromosome composition from both the parent into the progeny can be possible. But in the case of the asymmetric, the maximum chromosome composition is coming from the one parent. You have to remember, right? The maximum chromosome composition is coming from the one parent. From the another parent, it is having the least or zero inheritance can be possible. Such kind of the inheritance can be called as asymmetric type of the hybrid. We have another one we called as hybrid. Hybrid matlab cytoplasmic hybrid. There we are having the two type of the parents can be possible. For the production of the hybrid, again we are having the different techniques with the nucleus and without nucleus also. We are having the two culture. According to that, the fusion of the cell is going to take place for the production of the somatic hybrid. See, the somatic hybrid we are having now totally 6 type of the somatic hybrid can be possible. One is the interspecific hybrid, another is the intergeneric hybrid, another is the interfamily, another is the symmetric hybrid, equal chromosome inheritance from both the parent into the progeny, another is the asymmetrical type of the hybrid, another is the cytoplasmic hybrids we have in general. So these are the 6 type of the somatic hybrid can be possible. So in this method, right, the sexual process is bypassed in nature. That is the one things we have, right? So apart from this, right, so for the production of the somatic hybrid, there are the different protocol, right, we have in generally. So common protocol which is involved in the somatic hybrid production, right? One is that isolation of the protoplast. Isolation of the protoplast is going to take place with the help of the mechanical method, and enzymatic method. With the help of the mechanical and enzymatic method, isolation of the protoplast is going to take place from the different plant, either can be tomato or potato. Mechanical matlab with the help of the mechanical activity, right? So there is isolation of the protoplast is going to take place. Another is the application of the enzymes also. But among these two methods, enzymatic method is very common, is quite common compared to the mechanical method. Once after isolating the sufficient protoplast, then at the laboratory, at the in vitro condition, there is a fusion of the good protoplast is going to takes place. So in order to make the fusion or contact between the two protoplasts from the different plant species, we are using the different kind of the chemicals in the laboratory. Chemicals method, another is the electrical method. Even with the electrical treatment also, there is a agglutination or contact of the two protoplast culture in the Petri plate can be possible. Even with the chemical also, chemical application, there is a agglutination or contact between the two protoplasts in the Petri plate can be possible. Once after making the content, then we are having the selection of the hybrid within the given Petri plate. For the given hybrid, then there is a regeneration of the hybrid is going to take place. So in order to make the regeneration of the hybrid in the given Petri plate, we are applying the various kind of the media we call as MS media. Another is agar solution. Agar solution. Sometimes it can be application of the axins, and gibberellin also can be done to make the regeneration of the cell in the given Petri plate. It is. Then after the regeneration, whatever the plants will be developed, that plant is subjected to the characterization of the hybrid plant will be there. See, this is the common protocol we have in the development of the somatic hybrids here, right? So with the help of this technique, right, again I'm saying that because our fundamental objective in the plant breeding is to make the improved variations in the biological population, right? From the molecular breeding side, somatic hybrid is the one of the technique. Through that also improved variations in the biological population can be possible, right? So these are the few things we have, right? Thank you.